My name's Allison Jones. It's a pleasure to be here today. I work at the Institute of Women's Health at University College London, and I'm going to tell you a little bit about our new womb cancer test, and hopefully it will be applicable in this setting. Just to tell you a little bit about who we are. So the group is led by Martin Whitwenter. He is a gynae surgeon who has a very long track record in developing tests for risk prediction and early detection of women's cancer. Adam Rosenthal, who many of you will know, who's one of the UK's most experienced gynae cancer screening experts. Then there's me. So I'm leading on the laboratory logistics at the lab. My colleague Jonna, who's with me here today. And we have another gentleman called Dr. Mally Scott, who is specialising, has his PhD is specialising in Lynch syndrome womb cancer surveillance as well. So just a little bit about what we actually study. So there's been a lot of talk about DNA, and obviously the DNA code is incredibly important, as you all know, and that is in fact typed out in the right way without spelling mistakes. Also, there's another layer to that called epigenetics. So epigenetics has profound effects on how cells behave in very much the same way as a DNA mutation does. It doesn't, however, rely on a change in the underlying DNA sequence for it to have an effect on a cell. So one of the reasons that we're here today and we're not all one big blob of, boring cells on seats is because of epigenetics, because our bodies are highly specialized and we have different specialist tissues and cells, and all these cells behave differently. They all have the same DNA story. However, they read different parts of the story, and that allows them to express themselves differently. So epigenetics is therefore really important for normal health and development. One of the most important epigenetic mechanisms is DNA methylation. It's the most well studied, and it's what we study at the lab. And you can think of this as a little addition on top of the DNA that acts a bit like a switch that can switch a gene on or off, depending on its context. So the application of epigenetics is twofold. You can look at it in terms of ways of developing tests for risk prediction. So the important thing to also say is epigenetic mechanisms are often reversible. So throughout our lives and throughout the exposures to our environments and our habits, and whether we exercise, and all these things have an impact on our epigenetics. And we can use that to try and risk stratify people in terms of their disease risk. The other most important thing today to tell you is obviously that when tumours develop, everything gets scrambled, and that includes the epigenome. And there are DNA methylation features that we can focus in on for specific cancer types to produce tests of early detection. So at UCL, we had a very successful pan-European program of research that happened between 2016 and 2020. Unlike most people who collect blood and analyze blood for these sort of means, Martin was very, very keen to collect many different tissue types, also because epigenetics is expressed in a tissue-specific way, but in particular, the collection of cervical smears. So we had a cohort of around 5,500 women, and each of these women were really generous in giving us access to their smears, a mouth swab, and a blood sample. And we've done a lot of work on developing various different types of early detection tests, including those for ovarian breast that are in the pipeline. But I'm going to tell you more today, obviously, about the womb cancer test. One thing to mention, obviously, is that a lot of research, and it's very valid research, has been done in such a way, excuse me, that you will compare the cancer cells from a tumour with matched normal cells. And then you can look at them genetically, compare them, see what the biggest differences are at the top, and say, let's focus in on these genes because they're more meaningful. With 4C, the strategy was different from the outset. As I said, we were collecting cervical smears. So we're not actually directly comparing the two the tumour and the matched tissue, which you would always have to obtain in an invasive means. We're trying to use this cervical smear as a surrogate for what's happening elsewhere in the gynae tract, and picking up tumour cells that are draining down, or even cell-free DNA from the tumours. And then we analysed, we used the Illumina bead array. Basically, it's the size of a microscope slide, and you can analyse about 8 samples on one slide. And each sample you will scan over 850,000 different DNA methylation sites. So it's an epigenome-wide analysis. So what did we develop as part of 4C that's relevant to here today? So we've produced a test that we refer to as the WIDqEC test. And it basically has been developed from the cervical smear a bit further on now, excuse me, so that we can actually use a swab instead of a smear, because obviously as Neil mentioned earlier, for some women, obviously speculum is quite uncomfortable, so we want to move away from that. It's a really simple test that can be done quite easily, PCR-based, looking at methylation-specific PCR at particular genes. And these genes are GYPSY1 and ZSCAN12. And this is very much like the scenario you would get during COVID, where you would take a test and then you'd send it to a laboratory and then you get an answer within a few hours. So the main bulk of what we've published on this test is to do with really the application in women who have symptoms. That is, they have abnormal vaginal bleeding. So we looked at cohorts of women attending a UCLH specialist clinic for investigations of abnormal vaginal bleeding as a flag for womb cancer. Normally what would happen is the first line would be a TVS, a transvaginal ultrasound, to look at the thickness of the endometrium and to look for focal lesions, etc. And then that would be used to triage women into whether they have an invasive test or not. And the problem that you have with TVS is that it's not very specific. It's not very good at ruling out the women who don't have the disease. So this, I'm not going to present all the data from the paper. I'm just going to show this kind of more accessible modelling example of that data. So this is the scenario with the TVS, performance of TVS at 4 millimeters thickness, as to whether if the thickness is above the 4 millimeters, then the woman is considered to have cancer and therefore have the invasive hysteroscopy and biopsy. And you can see that there are about 28 false positive women there who unfortunately had to endure those often obviously painful procedures just to rule it out. But with our test, you can see that greatly reduces the number right down to about two to 3 women in this scenario. So this is great. But what else do we know about it? So there is other stuff going on at the moment. We've got some papers that are under review right now. But what I can tell you about the test also is that it does detect both serous endometrioid types, so both good and poor prognostic types of womb cancer. The test is highly correlated in a small cohort of around 120 women who took their own swab before they had investigations by a clinician who then took a second swab themselves at that time. So we wanted to see the performance of the patient versus the clinician, and that was highly correlated. And ethnicity, importantly, doesn't impact the performance of the test, because TVS, for instance, can be a problem in some black women because of prevalence of fibroids, and views can be distorted. So we know, I mean, obviously we've heard today that there are issues with womb cancer surveillance, and there's not really a standardized, focused program in place, unfortunately, but women are obviously offered it and offered hysteroscopy and biopsy annually if they want that. The thing about the WIDqEC is that it is a simple vaginal swab test, and it could be performed by women at home. which could be quite beneficial, particularly for women who have to travel from far places to get their annual surveillance. A recent retrospective study by Mali, who I introduced earlier, had showed that whilst what's actually offered at UCLH is annual hysteroscopy and biopsy, whilst pre-invasive disease can be detected quite often by the time the woman has a hysterectomy, there is often concurrent, more malignant changes. So what we're trying to do at the moment is understand more about our test, because we know that it detects endometrial cancer stage 1A, grade 1. But we also need to understand what's going on in the pre-invasive lesion stage. So we're investigating that at the moment. Moreover, Mali's retrospective also showed that interval cancers do occur. So these are cancers that are happening between the screening appointments. And the simplicity of the WIDqEC would hopefully mean that you could choose when you wanted to have that test. So maybe you could have it 3 times in a year, for instance. I mean, obviously, all of this has to be, these are all very hypothetical comments. And what we really need to do is some kind of UK-wide trial. And I know that, please don't take this for what it is. This is back of an envelope design, and you're the experts, a lot of you here, and know this field very well. And we'd be willing to absolutely collaborate with a trial if you were interested. We also have some CER UK funding for some Lynch syndrome focus groups, which we hope to hold one in London, and also perhaps even here. This is a great venue, by the way. So we can get some more information about your experiences and what you would be willing to do for screening, because we have to remember this is personal choice. You know, what might be right for one woman who is pretty anxious. She may not want to be tested or self-test quite as much as another woman, for instance. So it is something where we need to learn from you what is appropriate and what's needed. And we also obviously want to collaborate with clinicians in the field. So I'm going to leave it at that. We have got a little QR code there just for you to scan if you want to contact us. It's just a simple form where you can leave some contact details if you want to hear more about the research, or if you're interested in being collaborating in any focus groups. And that's it. Thank you.